not near the speaker. Okay. Good evening, everyone. Thank you very much for coming out on a typically Bristol spring evening. It's great to see everybody here and to welcome you to this, what has been an annual lecture but has been interrupted by world events, as we're well aware of. I'm Hugh Piggins. I'm head of physiology, pharmacology, and neuroscience here in Bristol, and it gives me great pleasure to welcome you all here this evening. This evening's speaker is Dr. Paul Chadderton, and Paul joined the school about 5 years ago. He trained initially in physiological sciences and then did his PhD at University College London, and then a postdoctoral research period there at University College, and then another one at Rutgers University in New Jersey. He returned to the UK to Imperial College, and then about 5 years ago he moved to Bristol, where he's now the Associate Professor of Neurophysiology. You can see that his talk tonight is about what happens next, and I'm really keen to find out what happens next. So without further ado, I welcome Paul to the lectern.
Okay, thanks Hugh. First things, let's see if this pointer works. Yes. Okay, so hi everyone, thanks for coming. My name is Paul. I'm a neuroscientist, and so the aim this evening is something which I actually bizarrely don't get to do that often, which is kind of ramble a bit about how I think the brain works. Okay, so that's the plan for tonight, and with the aim that you don't necessarily need to know anything when you come into the room. Okay, so I'm going to try and make a case for how I think particularly this part of the brain here, this part of the brain here, which is called the cerebellum, which I work on, what its function is, and hopefully you can make your own judgments about the ideas I present. So let's go. Okay, so we're going to start with a proposal. Okay, so we're going to start somewhere. So the proposal I want to start with is that the cerebellum is a part of the brain which its role is to make predictions about the consequences of future events. So the cerebellum makes predictions, and it does something with those predictions. Those predictions are used to control the actions of the entire nervous system, and indeed the whole body. Okay, so today's question is really, how does the cerebellum shape the future? All right, so what you're going to notice as we go through, because I was doing a public lecture, thought we needed high-quality images, so I signed up for lots of free trials where I could get 5 free high-quality images, put my credit card details in, and then just have to remember to remove them at the end of the period. But this is the first high-quality image, and so what we're thinking about here is predicting the future. Okay, so actually, if we want to predict the future, how are we going to do that? Okay, so we're going to start again simple. I'm going to tell you straight away how we predict the future, and then we'll go back. Okay, so the first thing, I'm sure you've heard this. This is a maxim which exists out there. Yeah, the past predicts the future. Okay, so that's something. So events in the past are very helpful for us in order to make predictions about what's going to happen when we're going forward. And then we can actually break that down a little bit. So I want to break this down into 3 steps, okay? So to predict the future, I actually need to know 3 things, okay? Only 3 things. The first thing is, where am I now, okay? So I need to be able to place myself in a certain position, and that position could be in any dimension that you would like to think. Then the second thing I need to know is then where I'm actually going. So in what direction am I moving? And then the third thing is to feed in this idea, how did things work out in the past for me when that happened last time? So we've got these 3 points here, and what I'm going to show you is how the cerebellum, this structure in the brain, at the back of the brain, is able to integrate these 3 different points as we go forward. All right, so before I do that, I need to give you some important background about the cerebellum. Okay, so first of all, what is about the cerebellum? So I'm going to start. The first important fact with the cerebellum is that the cerebellum is really old, okay? So if there's any proper signed up evolutionary biologists in the room here, I'm going to really embarrass myself. Yeah, but this is a dendrogram which basically explains how all animals with backbones have actually evolved. And what we can see is that basically all the animals from cartilaginous fishes upwards actually have a cerebellum. So what that means is that these things here, which are called lampreys, otherwise known as vampire eels, they don't have a cerebellum. But when we get into the realm of the fish, which have jaws like sharks, then these are animals that have cerebellum. So one of the things that I was fretting about a little bit when I was coming up with this lecture, I suddenly became very anxious as to whether dinosaurs had a cerebellum. Now again, that shows my level of evolutionary biological sort of base knowledge. You'll be relieved to know, indeed, dinosaurs did have cerebella. So here we go. So this is from quite a recent paper, and this is a reconstruction, and we can see, indeed, there is a cerebellum in this dinosaur's brain. Okay, so phew. So why am I making this point? Okay, so the first thing is that if something's very old, very conserved, it suggests that it's doing something useful. Okay, so the first clue is that it's doing something useful and something which is of fundamental value. So let's get rid of the dinosaurs there. And so what we can see is now, if I show you what the cerebellum looks like for a range of different animals. And so starting here, keep pressing the wrong button, we can start with a mouse, and then we can move all the way up to the human. So you'll notice the scale here is actually at the, up to the 1 meter scale. So you're thinking, how does that fit into the brain? So this is my high-tech visual aid that I've got for you here. This is University of Bristol prop. This is a sheet of A4 paper, which has actually been previously used. And what I've done is I've folded it into a concertina here. So I secured that with paper clips. And what you can see is that essentially we can unfold this. Yeah? So what you may be familiar in seeing this structure here. This is what the cerebellum looks like at the back of the human brain. But if we unfold this, actually with the human, this is just a tiny fraction, yeah? So we can unfold it so that we essentially have this 2 meter long flat plate structure. Okay, so that's the first thing. Cerebellum's really old. The second thing that is important to know is the cerebellum is really big. Okay, it's really big, especially in humans. So this is an MRI scan of a brain. So this is, if you go into an MRI machine, this is almost sort of sliced quite close to being down the middle of the brain. And within the average human brain, we have about 86 billion neurons. Okay, and we can divide this up quite well into almost 3 different areas. So we've got about 16 billion neurons in our cerebral cortex. So that's about just under 20% of all the neurons are existing in the structure here. And cerebral cortex is actually this, you see this grey sort of swirling pattern across here. You're probably familiar with that. And then actually in the cerebellum, we've got 69 billion neurons. So we have actually almost 80% of the neurons in the brain are residing here. And quite surprisingly almost, is that within the rest of the central nervous system, we actually have less than 1 billion neurons. So we've got this distribution going on here. And then the second thing that I want to point out to you is that there is a relationship between the number of neurons we find in the cerebellum and the number of neurons that exist in the cerebral cortex. So what we've got here is, don't worry about these, I don't even know what these words mean either, okay, but these are all different animals, let's just assume, yep. So for every different animal, we can plot the number of neurons in their cerebral cortex and the number of neurons in the cerebellum, and you'll see that what we have then is this very nice linear relationship between the two. And you'll notice the one thing I do know here is H is up here for human, okay? So in the human, we have essentially what we have is an expansion. We might be familiar with the idea that we have an expansion in the size of our cerebral cortex compared to other animals. But the important thing I want you to notice and be aware of is actually this expansion in the cerebral cortex is matched by our expansion in the cerebellum, okay? So when we see relationships like this, they can sometimes then begin to indicate to us that actually maybe these two things might be related to each other. Maybe there is a sort of codependency. Okay, so my first proposition then is actually that the cerebellum and the cerebrum, which is this area here, they've actually grown together. So they've evolved together. And so in the human brain, most neurons are found in the cerebral or cerebellar cortex. And the word cortex actually means, it's actually from the Latin, it means outer, actually from the Latin word for bark. So it's these outer layer structures of the brain which have very much overexpanded in humans. And we're familiar with this shape because as they've gone larger and larger, in order to be able to fit them into the cranium, then the sheets have started to become folded. So what if we then unfold these structures? Yeah, so this can be this will be helpful for us in a moment. So if we unfold these structures, then this gives us an idea of actually the true size of these two areas. So this is actually proposed by actually a very eminent neurophysiologist, David Van Essen. He pointed out that one cerebral hemisphere is about the size of a medium pizza. Okay, so within the cranium, we've got two hemispheres, cerebral hemispheres, and then we've got this rather chic Siciliana-style pizza, as you can see, this more oblong shape, which corresponds to the cerebellum. Okay, so we've got these two cerebral cortices, regular crust. The average thickness of the cerebral cortex in a human is about 2.5 millimeters. And then in the cerebellum, we have this super thin crust. It's actually like a millimeter. It's one of those crispy, I think it's called like a Leggera pizza or something at Pizza Express. But essentially, we've got these two flat sheets which are where most of the neurons are existing in the brain. And again, they seem to be kind of relating to each other in some way. Okay, so we're going to come back to that later, yeah? So now let's focus in then a little bit about actually the structure of the cerebellum itself. So if we unfold this highly folded organ, then we can produce this flat sheet here. And then once we have this sheet, what we can see if we look under the microscope is actually that there is this very simple repeating structure that we can observe. So if we just then take now one little slice through the pizza, then what we can see is that we have essentially the same organization all the way through. So what we have at the base is this very dense layer of very, very small neurons. And these are actually the most numerous neurons in the brain, the granule cells. And then sitting on top of those cells, we have these huge, beautiful neurons called Purkinje cells that look kind of, they either look like trees, they look like fan coral. What you can't see in this projection is that they're flat, but we'll see later. And I say, as the cerebellum has grown, all the way from these very old animals, well, very, very early in evolution, to now, this structure has been preserved. So we often describe this cerebellum as having essentially a crystal structure. It means something a simple repeating motif that is repeated again and again. Okay, so again, we're going to come back, so you can remember this if you like, but we're going to come back to actually this structure of the cerebellar cortex a bit later. All right, so summary part one. These are the points I'd like you to take. First of all, the cerebellum is old and it's conserved. So when we see that, that suggests that it's doing something fundamental. The second thing is that in humans, we're probably all familiar, or most of us are familiar with the idea that our cerebral cortices are expanded, but this expansion is matched also with the cerebellar cortex. And so this suggests that these two structures may be working together. They may be sharing tasks together. And the last thing is, if we can slice through this structure and see that wherever we look, it looks the same, that suggests to us it's doing one thing. In all cases, one thing. So we call this then a universal transform. So it doesn't matter where you go within the cerebellum, the thing it's doing is always the same. Okay, so what is it for? All right, so if people were going to jump out, if I was going to, I've blown it now, but if people are most familiar for the cerebellum being involved in movement. And this is actually comes all the way back from, I think, 1807, where people removed the cerebellum from pigeons and discovered that these pigeons had all sorts of motor defects. And then, during the First World War, there were many people who suffered shrapnel injuries. And if the shrapnel injury was at the back of the brain, then there were, again, a very characteristic set of issues. So I'll just show you, first of all, that we can see that we can hear this.
The cerebellar gait is characterized by a broad stand and by a wide staggering quality to it. People will tend to fall towards the side of their illness, so if the illness is in the cerebellar hemisphere on the left, they might fall in that direction. When asked to stand still, their trunk may sway like this, and that is called titubation. And obviously they would have problems with all the other cerebellar tests. One caveat.
Okay, so that's, you don't even need to really hear what he's saying. I just wanted you to see the kind of, there's essentially some quite characteristic movement issues that you can get with cerebellar defects. So if there's a problem in the cerebellum, and you might most commonly see that these days if someone would have a stroke at a particular place at the output of the cerebellum. So in this case, you get something called ataxia. So that means uncoordinated movement. So you have a loss of the ability to coordinate all of the limbs when you're moving. The second very common thing is something called dystonia. So these are different kind of muscle spasms that can occur, and they essentially some muscle will go up into a lock, and it won't then unlock. The other very common thing is to then have issues with balance and actually eye movements and fast movements. So it really has these quite striking effects on movement, but unlike the cerebral cortex, for instance, we don't see a total paralysis. We don't see a loss of, absolute loss of function in a spot. So it's a different kind of characteristic of loss. I don't need to go there. Okay. So that's kind of what's famous, yeah? And this was sort of really all the attention was been put onto this, I'd say probably up until the 1980s at least. Cerebellum was famous. It's this thing at the back of your brain, and it does movement, okay? And gradually, over the past 30, 20 years, we've come to see that actually it's involved in a lot more than that. So this is a paper from 1991, first of all, which actually was one of the first to really nicely, clearly characterize a non-motor defect in someone who had a damage in their cerebellum. So what they were asked to do, this is like a word association game, you're given a noun, and then you have to come up with a verb that associates it, yeah? So you're given the noun tree, and then you have to just say whatever, the first thing that comes into your head, what's a verb that goes with that? And so most commonly, you're going to climb a tree. Most commonly, a dog is going to bark. But in this patient who's had this right cerebellar lesion, they actually just totally did the task wrong. Yeah, so they asked for tree, and they end up saying tree house. They asked for dog, and they say cat. So there's something now wrong in their cognitive process, their thought processes, which are being messed up. And gradually, this has become more and more kind of pieced together, characterized, into something which is known as cerebellar cognitive affective syndrome. So cognitive being related to thought and affective related to mood and emotion. And so as well as having these disruptive thoughts and not being able to think clearly as effectively, also noticing that there can be personality changes. So things like blunting of affect, so sort of loss of emotional responsiveness, disinhibition, or inappropriate behavior. So here, we're going beyond purely the realm of movement. Now we're moving into the realm of thought and emotion. So this is just an example of someone who actually had then a very temporary inflammation around their cerebellum. So whilst the inflammation was present, their cerebellar function was impaired. So they were asked to draw, there's this there's this family of complicated drawings which people are asked to actually copy. And so this is just the same patient, and you can see, this is whilst they have the information, this is 3 months later. You can see that actually the sort of geometric organization is much better. It's returned after the information has come down. Okay, so that's thinking about movement, and then that's thinking about thoughts and cognition. Then really recently, it's become clear that actually these aren't the only things. And so actually cerebellum could be involved in all sorts of body regulation processes. And this actually, it's one of the ways this became obvious was looking at a particular group of individuals, and these are people who have a genetic disorder called Prader-Willi syndrome. And Prader-Willi syndrome is associated with a range of different symptoms, characteristics, but the one which is important for us here is hyperphagia. So that's just overeating. Okay, so if people with Prader-Willi syndrome, they're often obese, and it's hard for them to regulate their appetite. And so if this is not me now, but recently, it's a paper from the United States where they looked in the activity in the brains of Prader-Willi syndrome individuals and compared that with controls and looked for any areas which are different in terms of the response to food. And what they found actually, and so the way they did this was to put the individuals into a scanner, and then they will show them either food images or pictures of dogs that look like muffins, but not enough like muffins to make them hungry. And you basically compare then the response to the food image versus the non-food image in different situations, either basically when they're hungry, when they're fasting, or when they've just eaten. And what you see in normal patients is that you look at the yellow here, there's a difference in the level of activation in this area here, basically a little area at the bottom of the cerebellum, and this is messed up in the Prader-Willi syndrome patients. So this is then a clue that actually cerebellum is also involved in the impulse to eat. All right, so part 2 summary. What can we learn from looking at these facts in terms of what we actually think cerebellum does? So what we know is that damage to cerebellum can produce deficits in motor, movement, cognition, thought, emotional, and homeostatic behavior. Now, homeostatic means essentially keeping things as they are. That's what most of your body is doing most of the time. Okay? It's keeping your body warm, it's keeping your body oxygenated. So what this suggests is that this structure is a generalist. Yeah? It's really involved in all sorts of different functions. It's got a hand in everything. And different regions of the cerebellum do play roles in different behaviors, but with less specificity with the cerebral cortex. Now, I'll explain what I mean by that. Essentially, what we can do if we put a human into a scanner, we can look at which areas of the cerebellum light up in different situations. Maybe we'll ask them to do motor tasks, or we'll ask them to do cognitive tasks, we'll ask them to do emotional processing. And what we see is there is some sort of preference for certain areas to be active in different situations. But these are big areas, big landscapes, which are devoted to these different areas. And the thing which I'm not showing you here is that also in terms of where each part of the cerebellum is getting its information from, it's also sampling it very broadly as well. So looking at here in summary is that we've got a structure which seems to be playing some kind of, again, general role in a range of different processes. All right, so that's what we know from looking at it, doing a bit of evolutionary biology stuff, also looking how patients with cerebellar damage are different from normal individuals. What can we actually learn now actually about the function? So this is kind of the sort of thing which goes on in my lab, where physiology is essentially the discipline of trying to understand function, the function of living things. And so let's actually go in and see if we can understand something about function of cerebellum. All right, so we've come back here to this picture here. This is the cerebellar cortex, one slice through it. And you might be familiar with the concept that we sometimes describe we call brain circuitry, okay? So that implies that different structures are connected to each other in some kind of organized way, yeah? And so this is an example of a brain circuit. And what you need to know about this brain circuit is quite simple, yeah? Information is arriving in here, and then it gets sent up here, and then it goes out in the middle, okay? So it actually arrives here, gets transmitted up here onto the green cells, and then the green cells send the information out, okay? So we've got two things to worry about. So we've got these little cells that live down in the input layer, called the granule cells. Very, very small neurons, but there's little sort of bubbles. And then we've got these big neurons, the Purkinje cells. Yeah, and these are the cells that send the signals out of the cerebellum, all right? So what you can see here, we call this then the dendrite. This is where the Purkinje cell gets all of its information, and then what you can't quite see here, it's a little bit cut off, it will send its axon, is this little wire out of the cerebellum here, and this is just a recording pipette, which is used to fill the cell. So we're going to look at how these different elements, what they're actually doing, okay? So we're actually going to go in and record the signals there. And we're going to do this, we want to do this actually in a situation when something is happening, yeah? So there's many different situations we can do this. One way in which we do it, in my lab and in other labs, is to actually look in the part of the cerebellum which is involved in whisking. And the reason we do that, the reason we look at whisking is that we are using rodents, because that is an animal model where we can do quite detailed experiments, and we can use different sorts of molecular tools, but they're also the most simple animal that we can actually use. And then the reason we use whisking is because whisking is quite a complicated behavior, and it's somewhat analogous to the way that we use our hands. So rodents, rats, mice, other rodents, they use their whiskers to actually explore structures close to them. They can be quite delicate with their whiskers, and so this is essentially their main way of actually using of touch information. So what we can do now is we can see, so then the one other thing I want to say to you is, if you notice what this rat is doing, yeah, so this is an active process. In the same way as if I give you an object to explore with your hands, it doesn't just sit there in the palm. You use your fingers to actually make movements around the structure, and the rat is doing the same thing here. So as it's doing its sensing, this is an active process. So what we want to do is understand what are the signals in these different areas of the cerebellum which are present whilst, to begin with, the animal is doing a tactile behavior. Okay, so here we go now, we're going into the lab. Yeah? So we're showing you, we are electrophysiologists, that means we record electrical signals mostly, and we normally do this in cupboards, ideally cupboards underground, because then we don't have to worry about electrical noise messing up our recordings. And we like to record them on these things here, these oscilloscopes. So there's actually an animal off to the side, but this is a recording that my former PhD student did, Susu. And what you can see is when the mouse moves its whiskers, you can notice these electrical signals appearing on the oscilloscope. So if you're able to see them, it's going to do a little whisk in a moment. And you see those sharp downward deflections. So those are actually individual synapses that the neuron has being activated. Yeah? So they're receiving signals which are coming then into the cerebellum. So this is what the cerebellum is hearing, if you like, yeah, from the rest of the brain. And then we can ask, what actually is the cerebellum saying to the rest of the brain? So we can record now from one of these big green neurons, well, they're not actually green in the brain, these big neurons, the Purkinje cells. And again, if we do that, whilst the animal is moving its whiskers, in this case, this is the movement of these three whiskers here. What you can see is there's a changed activity. So that clicking impulse that you can hear is actually the electrical signals of the neuron, and it communicates with other neurons via these, they're like these little digital clicks that occur. And so you can hear that the activity of the neuron is changing as the animal changes, animal moves its whiskers. Okay, so I'm now showing you a horrendous graph, yeah? So it's like, eek, but don't worry, easy graph. I think this is a graph that everyone gets. So what we can do is, with that video, is that we can chop that video up into different periods when the animal moves its whiskers, and then we can basically plot, first of all, where the whisker is, so we can plot it here, and then underneath we can plot what the activity of the individual cell is. Yeah, okay, so the top trial here corresponds to the trial here. So what your eye should be saying to you is actually these two signals look really, really similar to each other, yeah? So, and actually maybe if I were to ask you to draw what you thought the whisker was doing on the basis of looking at what the neuron is doing, you might actually be able to have a crack at that, yeah? So actually the cells change the rate of their activity in a way that allows a readout of where the whisker is going to be. And it is going to be, because what we can do is we can look at the timing of when the neuron is active versus where the whisker is. And what we find is that actually, on average, these neurons are going to tell us about 50 milliseconds before where the whisker will be. We'll do that the right way around. So these neurons are giving us information about upcoming movements. Okay, so point one, there we go. Thing to take home, these neurons are reporting upcoming movements. Now, I said to you, cerebellum is not just involved in movement. Okay, so this is just a very recent example, which was collected by Danny in the lab, to show we can also see signals in cerebellum related to these other behaviors. So in this case, there is a mouse there, and the mouse is going to get his dinner in a moment. So I think Danny, that's you, Danny. Yeah? Danny comes in and puts some food down for the mouse, and the mouse will go and collect it. And if we record at the same time the activity in the cerebellum, what we see is that when the food is delivered, we start to see this increase in the activity of cerebellum. So it's quite a different sort of signal now. It's a much slower signal. And this is not actually relating to the speed of what the animal's doing or where it is, but it's actually, again, some sort of predictive signal that the animal knows that it's about to go and get some food. Okay, so cerebellar neurons tell us things about upcoming behaviors. That's the first thing that we know. The second thing that is quite special about cerebellar is that these neurons, these Purkinje cells, also get signals when behavior does not turn out the way that it is expected. So I'll just explain what's happening here. So this is data that Simon has collected, and what he's done here is he's put a fluorescent, so a glowing protein into the Purkinje neurons, and now he's actually using a microscope to observe what these neurons are doing, again, under conditions where the animal is able to move its whiskers. And what you should see, what you'd like to see is these little flashes, these impulses which occur. Let me just show that again. So in this case, we're in a slightly different axis, so each Purkinje cell is much flatter. But you'll notice, do you notice these flashes which are occurring up here? So these are actually signals which are very commonly tripped by errors, okay? And so we know this is then some actual classic data collected from, in this case, a primate when they're doing a task where they're having to move a lever. And what we see is that these flashing signals here essentially encode when errors have occurred. So when something happens to the brain that it's not expecting, we see these error signals present in the Purkinje cells. So we're encoding upcoming events, but we also have an error signal present. And then the third important thing that you need to know is that cerebellum can actually make stuff happen itself. So this is now a top-down view of what a mouse cerebellum looks like. So you see it doesn't have to be as folded up as a human one. And then if we just focus on one area of the cerebellum and we activate that area of cerebellum, what you see here is that we can make the mouse move its whiskers. So driving this one spot of the cerebellum is able to actually produce behavior. So these are the things then I want you to know about kind of how we think cerebellum does. So cerebellar neurons represent upcoming actions. This includes precise movements, but also other behaviors. Cerebellar neurons receive signals telling them if the right or wrong thing has just happened. So they're getting feedback. And the third thing is that the cerebellum can make stuff happen. So changing cerebral activity changes behavior. Okay, so now we know that, we can now go back to where we started, yeah? We can go back to this question of how do we actually predict the future. So, like I told you before, to predict the future, we need to know three things, okay? We need to know where we are now. We need to know where we're going, and we need to know how that worked out in the past, okay? So what are those three elements? How do we see those three elements in the way in which the cerebellum itself is behaving? So the first one is quite trivial, okay? So you've seen this video now. This is a recording, and what we do is we're recording from these little granule cells. We're seeing the signals they're getting. And what I didn't show you last time is what we can actually do then is that we can plot how many of these synaptic events are occurring at all of the different positions that the whisker can be at. And what we see is that there is a relationship between how many of these events occur, so the rate of activation, and where the position of the whisker is in space, all right? So basically, how activated the cell is, in this case, tells us where the whisker is, all right? So it's quite simple. There's lots of other parts of the brain that can do that, yeah? But this is step one. So step one says, I am here, okay? So that's great. But where am I going? Okay, so for this, we need to do, it's a little bit more complicated, but it means that I get to use a graph that I've made in PowerPoint, which is always really exciting that I can still do that. Okay, so we get a simple graph that we can do in PowerPoint. So what we've got is we've got two axes here. So we've got time and we've got position. So in this case, we're thinking about position of a whisker, but it doesn't need to be that. This could literally be anything. And like I said, we know where we are right now. Okay, so this is the present, and this is our position in the present. And then as we look out across the time horizon, we've got the future in front of us, and we've got the past, the immediate past behind us. So what we can do is a trick. Yeah? What we can do is we can look where we were very recently. Yep. So we know where we are now. If we could look where we were very recently, then we could maybe look at something like trying to come up with the gradient of the change of what's happening there. And then we can use that to propel it forward and make a guess about where we're going to be in the future. Okay, so essentially what we use is the very recent past to help us inform about the direction in which we are going in the future. All right, so how would we do that? How would the cerebellum do that? So the way the cerebellum does that is with a trick it's essentially a wiring trick. One way the cerebellum does that is a trick of the wiring. We're using these input cells, the granule cells. So I'll show you here. So again, remember that this is what our cerebellum looks like normally. It's all the folded up version. And if we unfold it, one of the reasons it folds so well in this particular axis is because each of these tiny granule cells sends its axon, its little emitter cable, straight up, and then as it gets into the region where the Purkinje cell is, it splits in two. And it splits, and it basically runs horizontally in two directions. So all of the granule cells are sending their axons in this direction here. So that really makes it really want to fold up very nicely. And so if we look in that axis now, what you can see is, this is a very old classic textbook, but this describes it very well, that these are the little granule cells that exist here, and they send their little axon, which is going to be the signal that is communicating with the Purkinje cell. It goes straight up, and then it splits in two, and it moves off in two directions. Okay, so this kind of anatomy, then we can use it as a little trick. So what we can say is, for a given Purkinje cell that's sitting here, okay, then we've got a granule cell straight underneath it. Yeah? So this granule cell is living right underneath it. It receives information about the position of a particular thing, where the whisker is now, and it sends that signal straight up, and then it sends its signal and it arrives onto the Purkinje cell. Okay, so that gives us information then about the present. But what about then this granule cell which is a little bit further away? Okay, so this granule cell also has information about position, but the information is now going to have to travel further to reach this Purkinje cell here. And this is a non-trivial distance, so this is a genuine delay in terms of the amount of time it takes for the signal to arrive. So what this means is the signal arrives a little bit late, but practically what it means is it's going to be telling us information about the very recent past. Okay, so this individual Purkinje cell is now privy to information about now and the spectrum of recent past as well. And so it can use this information to actually then make a prediction or essentially compute that gradient, that slope, and make a prediction about where we're going to be in the future. Okay, so step 2. Essentially, the trick of step 2 is that I know where I came from, and therefore I know where I'm going. Okay, so we've got a position, we've got a trajectory. So now, where is that trajectory heading? So this is step 3. Yeah? So how did things work out in the past? And this trick then relies upon these structures here. So this is now a microscopic image of just the top part of the Purkinje cells. So those, that tree-like structure. And what I want you to see is, if you zoom in, you can see these little bumps, yeah, these little nodules that come off here. And so the whole of this Purkinje cell is covered in these tiny little, what we call spines. Yeah? And so each spine is essentially the contact point for one of these granule cell inputs. So it's arriving there, and so each Purkinje cell is going to get, in the human, it's about 150,000 of these. So we've got 150,000 spines, and each spine hosts a synapse. Now, what's special about these synapses is not the only place in the brain, but many places in the brain. The strength of these synapses can be adjusted, and it can be adjusted in a way that then is maintained. Okay, so we call this plasticity, and long-term plasticity. It's something which is sustained over a period of time. So what we need to do is we actually need to train these Purkinje cells to remember, or for their synapses to be of the right strength. And so how does it do that? So this is now, this is a really cool paper from a group in Japan, and what we've got here is, what they're doing is they're imaging the whole of this surface of the cerebellum of a mouse. Yeah? So as much as you could look at the whole brain in one go, that's what they're doing. And then they're, similar to what Simon's doing, they're looking at these flashing signals which correspond to the error signals. And so as they're observing a mouse doing behavior, what you can make out these, maybe you can make out these little sort of little bands, these little stripes that are coming on in different places. But these signals are active all the time. Yeah? So these are the signals which actually change the strength of the synapses on those very small ends of the Purkinje cell. So these essentially are teaching signals. The teaching signals are going to change the strength until the behavior that the animal does is no longer producing strange results. Yeah? So it's like a permanent, well, it's essentially a permanent tuning process, but this, for most of us, this tuning process, most of it is happening when we're very young, so we're not noticing that it's actually taking place. But essentially what we're doing is we're adjusting all of the strengths of these different synapses using these teaching signals. So we adjust, the signal helps us adjust the strength. And then, essentially, this is what it will enable to do. So we'll train the Purkinje cell to have the correct strengths on its synapses, and these stored properties then essentially shape what the Purkinje cell, the signal the Purkinje cell will send out. Okay, so for a particular set of granule cell activity, they will produce the correct output, i.e. an output which no longer produces an error signal. And every time there's an error signal, then there'll be a readjustment again, and so we can keep this thing up to date. And so then how does this work? Yeah, so you might be most familiar with an example of how this works if you've ever been on the London Underground and you're feeling like you've been lazy all day, so you're going to use the escalator that isn't working, the one that they haven't bothered to turn on. Okay, so this is a really famous, it's called the broken escalator effect. But it's one that it's actually very hard to abolish. So as you step onto an escalator normally, you need to decelerate, yeah, as you do that, because this thing is moving. But when you do that correctly, actually then your vestibular information doesn't change. So you've slowed down, but your body-brain knows that you're not going to get the signal that you've slowed down. You're actually staying the same speed, yeah? So and that works normally. If you ever take a small child onto an escalator, you know it can be dangerous because the first couple of times they do it, they will facepalm because they haven't learned this association yet, yeah? But then when you go onto the broken escalator, there's this very, very strong expectation that you will not be feeling a deceleration. And so when that doesn't come in, it's extremely jarring and noticeable. Yeah, so that's one instance, but all of these physical relationships, non-physical relationships, social relationships, these are all connections, associations which we are building up all the time. Yeah, and in a way, the sign that they're working is when we don't notice them. Yeah? It's we only notice them when something is surprising or something that has been a certain way is no longer the way that we thought it should be. Okay, so some part 4 summary. Let me just check what time it is. All right, so cerebellum receives rapid information about the current state of everything. So obviously I don't have time to show you all the different kind of signals that it gets, yeah, but it's getting information about all sorts of everything that's really changing in the body. The cerebellum compares current and recent states to calculate the future direction of action, okay? And this direction and current state are matched to then what is the cerebellum's best estimate of what should happen next. And this estimate has been built up through experience and then through these changes in the synapses. And yeah, so there we go. So that's all great, yeah? So we could be happy with that. Now I'm going to burst my own bubble a little bit, yeah? And say, actually, well, the cerebellum is not the only part of the brain that does prediction, that predicts the future. Yeah? Potentially, that's what every single part of the nervous system is doing. They're just doing slightly different tasks, yeah? They do different jobs. So with that in mind, then actually, what might the cerebellum be doing which is really special, yeah? Which is different, which is why its structure has been conserved, why evolution has decided that it's a useful thing to have around. Okay, so I'm going to give you 2 examples, yeah? So the first one, another high-quality image here. This is a high-quality image of a ripple on a pond. So this is an analogy, yeah? It's always and for me, dangerous because there's no words on this slide, so I can be totally untethered, yeah? But the reason I'm showing you this slide is to give you the idea of how signals propagate, all right? So it's easy to understand. In a pond, if I drop a pebble in the middle of a pond, we know that there's a displacement that happens at the point at which the pebble goes in, and then there's also a propagation of this signal, yeah? And this is something which also happens then in the brain, yeah? And you may be familiar with brain waves, yeah? These waves that we can actually, electrical waves which we can pick up on the surface of the skull. So there is this propagation of different signals. So in this case, what we can see is that the pond or the lake is nice and calm and it's smooth, and there's a very smooth transmission of information, of the signal. But that's not the world we live in, yeah? The world we live in is one where things are constantly changing, there's all sorts of different things happening at the same time. And so this kind of organization here is kind of potentially challenged. So I'll explain to you a bit more what I mean. So if we go back to this video here, we can say that there are different rhythms in play, and there's sort of 3 we can see just in this context, yeah? So the different rhythms that are present are that there's the whisking rhythm, so there's the movement of the whisking, but then actually this rhythm is linked to the breathing rhythm of the animal. Yeah? And so there's a link between whisking and breathing. There's also a link between whisking and walking. So if I'm walking, I'm not now walking, I'm not whisking anyway, yeah, but when the rodent is walking, there is a relationship between the whiskers and their movement. So now we've linked together three things. We've got breathing, we've got locomotion, we've got whisking, okay? There are many more things going on at the same time. So it's the same for us. When we're walking around, our breath is relating to our locomotor patterns. And so you imagine now this presents a problem. Basically, the problem is that if we have a change in one system, then it has the potential to influence many or all systems. Yeah? And I hope you just intuitively get an idea of why this might be problematic. Yeah? The idea of chaos theory and the famous analogy of the butterfly flapping its wings in Brazil, and this tiny perturbation in one situation causing a propagation of events that leads to something totally unexpected. Yeah? In a practical case, for me, I'd probably just fall over. Yeah? But it could be caused by any sort of disturbance that's going on in the brain. So this is a real problem. Once you have a complicated system where lots of different areas are all talking to each other, yeah, so there's different signals, different competition, and what we call noise, yeah, so things that we don't necessarily expect. And this then is ultimately the consequence of if this is not an unregulated process, yeah, so we go from this smooth condition of signal transmission to this now turbulent thing here, where it's impossible to know what the hell's going on and how it started and where it's going. So this is what we need to avoid. So this may be one of the things that cerebellum is really optimized to do well. So I'll show you. So this is some work done by Ensor in the lab, and I'm going to freak everyone out now by showing you a model, a computational model. So everybody, duck. Okay, don't worry, there's wine coming soon. All right, so I'm going to shield you from this model with some emojis. Yeah, so we keep it simple, and we'll bring back the pizza analogy. Okay, but this is really, we can just understand this on a qualitative level. That's all we need to do. The idea is that we have these different structures in the body or in the nervous system which are specifically tasked to control specific aspects. Okay, so we have areas in the brainstem that control breathing. We have areas also these tend to be areas which are sort of very well conserved lower down in the brain that control whisking and control locomotion. And so all of these areas are actually interested in making their own predictions, and then those predictions enabling the right thing to happen. Okay, so they're all interested in that. But there is this problem that can happen where something unexpected happens in one area, and this can have consequences for the other. So the idea is that actually, because the cerebellum is in this position where it gets input from all of the different areas, it's this generalist structure. What it can do is it's able to actually learn what is the correct way in which these structures interact with each other. And when there is a situation where one element is about to push the other elements into disorder, cerebellum will correct for that. And so I show you we can represent that in a model. So this is actually, then if we play this model, we can actually explore that. So what we're going to do is we're going to look at the whisking oscillations, because the animals are moving their whiskers like that, and we're going to also look what's happening to the respiratory oscillation, yeah? So the breathing rate. And we'll do that first of all when the cerebellum is present, and then we'll turn it off, and we'll see what happens then to the signal. And so the top one is actually what's going on. The middle one is actually how synchronized or unsynchronized these two things are. So when it goes red, we've turned off the cerebellum, and what you see is that we very quickly have this loss of coordination between the two structures. Yeah, so they begin to just lose their ability to couple. Then we turn the cerebellum back on, and the cerebellum essentially is able to bring these back into sync. So the cerebellum in this case is providing the correct commands to both of these different centers to correct online what might be what would otherwise be leading to this disordered state. So that's one of the things cerebellum is doing. This is potentially a very general thing it can be doing for any independent area. Then there's another thing which cerebellum is highly likely to be doing. Yeah, so we're going to go now back. So this is another model. So I'm going to quickly shield again. This is a model now where we look specifically to the interactions of the cerebral cortex and the cerebellum. And the cerebral cortex, we know the cerebral cortex is very important. Yeah? And if you destroy the part of the specific areas of the cerebral cortex, if you destroy them, they can really cause debilitating loss. But cerebral cortex has got its own problems, okay? So with the cerebral cortex does not get information about the consequences of what it's done very quickly, okay? So if the cerebral cortex, if a command is delivered by the cerebral cortex, it takes a long time for it to find out whether that, the consequences of what it did was actually what it wanted to do. So there's a trick, yeah, where the cerebellum can come in, and what the cerebellum can do, because the cerebellum is so good at making very fast predictions, if the cerebral cortex, if an action is triggered in the cerebral cortex, what the cerebellum can do is provide a very fast estimate of what the result of that action will be. And the cerebral cortex can use that essentially as a compensation to actually get in the real feedback. So again, don't worry about all of the different bits and bobs here, but what I would say to you is actually what this stops, this external feedback essentially means that the cerebral cortex doesn't always need to rely on this slow signal to come back. Instead, cerebellum can actually help out. And so the way I like to think about this is essentially these two structures, they may be doing sort of from the very, very zoomed out way, similar things in that they're trying to make accurate predictions about the future, okay? But they're working at slightly different rates, yeah? So the cerebral cortex is working at a slower frame rate than the cerebellar cortex. Yeah? That has certain advantages. So actually, cerebral cortex, if I'm going to ask you about where you're going to go for your dinner after this talk, yeah, you're going to set off a cognitive chain there, which is going to be highly dependent on your cerebral cortex and your ability to take information, to plan forward into the future. What cerebellum is really good at doing is stitching together these sorts of events. So we can think almost about having two different frame rates in the brain. We've got the slower frame rate of the cerebral cortex, and then we've got this very fast predictive frame rate of the cerebellum. Okay, so summary of that. So here we go. So cerebellum communicates with all other brain systems. It uses incoming information to learn how different systems interact and to correct these interactions online. It operates at rapid speed. Now, I didn't show you, but actually cerebellar neurons, they fire much higher firing rates than cortical neurons. So if you think that you need that ultimately, like when the neuron is actually signaling, that rate tells you something about how often the signal can be updated. That tells us the cerebellum is operating at speed. And so what it's very good at doing is coming up with these fast predictive feedback, and it can provide this to other brain regions when they don't have access to that information themselves. And then later, it joins it all back up together. Okay, so we're almost finished now. I think I heard some bells or something. So we're going to finish with the summary pizza schematic. Okay, so we've got these different structures of the brain. And I really got carried away with this. So we've got left cerebral hemisphere, right cerebral hemisphere. We've got cerebellum. These are the subcortical nuclei, doballs. The thing which is missing is the chi strings, which connects everything together. And so what's actually going here? How do these different structures working? How are they talking to each other? Okay, so we just picked one little spot on the cerebellum. Okay, it's getting broad information from different areas about what is happening now in those areas. Plus, it gets very fast feedback from the periphery about what's actually going on in the outside world. So it uses this information then to provide the update, the prediction of what actually should be happening next. And depending on where you're on the cerebellum, this signal will be going distributed to the relevant areas in the brain. So here we go. So this is my closing thoughts. Yeah. So the whole brain, we can think of the whole brain as a prediction organ. Yeah, we can even think, we get into this, something which really changed my way of thinking is like, we can even think about the motor neuron as not providing a motor command, but it's providing a prediction. And the sensory signal coming back It's not a sensory input, it's a prediction error. So we can think about the whole brain in this way. But the cerebellum is special in that it is optimized to make widely informed predictions across short timescales. The other thing I didn't tell you, which is a real big difference between the cerebral cortex and cerebellar cortex, is that the cerebellar cortex is designed, information goes in and it goes straight out. There isn't really an opportunity, apart from this special parallel fiber signal, for it to hang around. Whereas cerebral cortex is full of neurons connected to other neurons, connected to other neurons, connected to other neurons. And so information can just stay going round and round and round in the cerebral cortex. Whereas cerebellar cortex, it goes in and goes out. So that tells us that what it's probably not going to be good at is predicting things in a very long way in the future. But that's the trade-off, yeah? So we get this, instead, rapid answer about what happens next. So at longer timescales, cerebral cortex takes center stage. But even in this case, cerebellum is essential to be able to stitch together all of the sensations, impulses, actions, and thoughts that are going on in the brain. So it's actually one thing that will potentially gives us a complete, a holistic sense of self. Okay, so this is my last slide. So you've seen some of the people who have done this work, and then I just wanted to flag a few of collaborators, Connor, Rui, and Leah, and Ellie, who has done a lot of great work in the lab, but there's no way I could jam it into the lecture. And that's all I have to say, so thank you for your attention.